Breakdown of glycogen is some of the most important aspects of keeping blood glucose in check. And so we're going to discuss a little bit about how glycogen metabolism is controlled. Now, our liver and muscles are our main stores for glycogen. And remember that glycogen is our body's short-term solution for energy shortages. And our long-term, of course, is storage in fatty acids. But fatty acids take a while to break down. They have to be transferred to the liver in order to be processed, and so it takes quite a while to process them out. Glycogen is a much more available source locally in muscles and a large supply in the liver to be sent out to bring blood glucose up when it's needed. So this whole system feeds out of the hexose monophosphate pool. Of course, this is fed by glucose coming into the cell in terms of hexokinase. Phosphorylation brings it in as glucose 6-phosphate, and of course then it can isomerize or mutate into either fructose 6-phosphate or glucose 1-phosphate, respectively. Glucose 1-phosphate is used, of course, as the starting material for glycogen. It is first combined with UDP glucose using the enzyme UDP glucose pyrophosphorylase. And that is an enzyme that is named for the reverse reaction, but it essentially takes glucose 1-phosphate and makes UDP glucose out of it using a UTP. And that reaction ends up cleaving 2 phosphates off. Really, it's a pyrophosphate, but those break down into, of course, two phosphates. PPI goes to 2PI with a delta G naught of negative 19 kilojoules per mole. So once we have UDP glucose, we can use that for a lot of different things in the cell. You can use it to glycosylate proteins, or use it for epimerase type enzymes, or a whole bunch of different things. Use UDP glucose. So we're not yet dedicated to making glycogen at this point. The place where we're actually dedicated to making glycogen is glycogen synthase and glycogen phosphorylase. Those are our flux controlling steps. So I'll underline those guys. And then these are counteracting. One of them is for the synthesis, the synthase part of making glycogen, and the other one is for the degradation of glycogen, the phosphorylase piece. Now, glycogen should be stored when you have a lot of extra energy around to store. So a key concept here is controlled by our ATPs and ADPs. When ATP is high, that means we have a lot of energy, and we probably don't need to be breaking down glycogen to liberate extra energy because there's plenty already. So ATP directly represses phosphorylase by acting as an allosteric inhibitor. On the other hand, if we have a lot of ATP, that should be a signal to our body that instead of breaking it down using phosphorylase, we should probably store it using glycogen synthase. So ATP has the opposite effect on glycogen synthase. And we'll see that same kind of thing happening in glycogen metabolism as well as in glycolysis, and we'll see that with hormonal control as well. Now, if you have AMP building up, or ADP, but AMP is even more powerful, that means that you're running out of energy. And so AMP should be activating a breakdown, because that means your energy level is low. You should probably break down your short-term reserves so you can get more energy, right? Make more ATP. The whole purpose of liberating glycogen is so it can go in, for either into glycolysis through F6P to make more energy, or you can use it to send out of the cell in the liver and modulate blood glucose. Now, the same thing happens over here for glycogen synthase. If we have low energy, we probably don't need to be storing it in glycogen, and so that should repress glycogen synthase. Now, a couple other interesting things here. If we have a lot of glucose 6-phosphate, that means that our hexose monophosphate pool is backed up. There's a lot of stuff here, and we don't want to keep filling it up. It's going to overflow. And so glucose 6-phosphate actually represses glycogen phosphorylase here. And if that means that our pool is full, we're going to probably want to turn on glycogen synthase. Glucose 6-phosphate represses the breakdown because the pool is already full, and it favors storing so you can empty the pool out. So that means that you have a lot of stuff built up here. You need to put it somewhere. Let's put it in glycogen. That's an easy place to put it. So that is a level of cell autonomous control. That is levels of different intermediates or energy cofactors or energy substrates are going to influence either the production of glycogen or the breakdown of glycogen by acting counter to one another at this flux control point. Now, kind of building off of this, there's an additional level of override for the endocrine system to tell the cells what to do even if they don't really want to do it. And so, for example, in the liver, the whole purpose of the liver is to send out blood glucose when you need it. Liver is a great store of glycogen, and so you need to liberate your glycogen reserves. And so what's going to happen here is signaling either by adrenaline, or epinephrine of course, or glucagon, which is a signal from the pancreas that says that a blood glucose is too low, will trigger the liver. These receptors being triggered turns on the enzyme adenylate cyclase. Adenylate cyclase turns ATP into cyclic AMP, the second messenger, and that activates a key regulatory enzyme called protein kinase A, PKA. So whenever cyclic AMP is around, PKA is on. Now if you want to look back at the old glycogen or the glycolysis regulation videos, you'll remember that PKA the flux through glycolysis is modulated by these hormones. And PFK2, for example, in the liver is turned off. Now, turning off PFK2 means that we go down in fructose 2,6-bisphosphate. Fructose 2,6-bisphosphate is a key activator for PFK1, which is the flux control step into glycolysis. So if PFK2 is off, PFK1 is off, and therefore glycolysis is off. Now another thing that happens is PKA turns off pyruvate kinase. Pyruvate kinase is at the end of glycolysis. It's the flux control out. So flux control in and flux control out of glycolysis in the Krebs cycle direction are going to both lead to glycolysis being turned off. Now that's important because the liver doesn't want to soak up all of its glycogen reserves as soon as it's triggered. So the other part of this is that protein kinase A will turn on an enzyme called phosphorylase kinase. Now, look at the name. Phosphorylase kinase is a kinase that phosphorylates phosphorylase. Phosphorylase, again, is the enzyme that breaks down glycogen. So when this thing is on, phosphorylase is on, and glycogen breakdown turns on. So glycogen is going to fill up the HMP, and it won't go into glycolysis. And that's key because that means we can ship it back out. Remember, that's what the whole purpose of glucagon is. Now, if you are in fight or flight mode, this would be also a good thing because you want to bring the glucose up so you can run away from a bear or lion or whatever you happen to be running away from. And so having glycogen break down and not be sucked right into glycolysis is key. Now, protein kinase A is also smart because it turns off glycogen synthase. So rather than just getting stuck in a cycle where we break down glycogen and then we just make more and then break it down and make more, PKA turns on breakdown and it turns off synthesis. And so synthesis is off, breakdown is on. The final thing PKA does when it's activated is it turns on the inhibitor for protein phosphatase 1. Now, that is the counteracting partner of PKA. This thing undoes all the phosphorylations that PKA does. So whatever PKA does, PP1 is going to undo. So that's very key to understand that. So that's liver. All of these outputs mean glycogen builds up in the cell, it doesn't get sucked into glycolysis, and you can send it out for the rest of the body to use. Now, on the other hand, in muscles, we don't have a way of shipping out glucose. We don't have glucose 6-phosphatase, and our receptors, our glucose transporters, are too high affinity for really effective pumping out of the cell. They're going to be saturated all the time, and we're just going to end up pumping one direction, and you can't even get the glucose 6-phosphate off. And so the only thing muscles respond to is epinephrine. Epinephrine turns on adenylate cyclase, that turns on PKA, that turns off its inhibitor, of course, and its counteracting counterpart. Now, in cardiac muscles, FBPase2, the phosphatase that breaks down fructose 2,6-bisphosphate, is turned off only in heart muscle. And so this makes your heart race. Your glycolysis turns up because F26BP goes up, PFK1 turns on, and glycolysis turns on. So in cardiac muscles, glycolysis turns on faster. In skeletal muscles, it's already on all the time. It doesn't need to be controlled. The other thing it does, of course, is one cyclic AMP is produced by epinephrine, PKA turns on, phosphorylase kinase turns on, so we break down glycogen, and we turn off synthesis. So in this case, glycogen breaks down and it goes straight into glycolysis, which is why your muscles need it most. So your heart needs to beat faster, your skeletal muscles need to run, and so this whole thing works really nicely for overrides of control. So glycogen breakdown and glycolysis are integrated by the same signals. Start with epinephrine to glucagon and you get key changes in how the cells are responding. So here's the big picture about glycogen. Here comes our ligand, it's either epinephrine or glucagon. That is going to trigger adenylate cyclase. Okay. Adenylate cyclase makes cyclic AMP from ATP. Cyclic AMP relieves a repression on protein kinase A. Protein kinase A then phosphorylates kinase, so it's on, and it also phosphorylates PP1 inhibitor, which means it is on and represses the counteracting phosphatase. PKA phosphorylation of phosphorylase kinase then activates glycogen phosphorylase, which is now on, and so a net flux is to break it down. Now, PKA also directly inhibits glycogen synthase. So that means that we can't go down this pathway anymore. All of the net flux from glycogen goes straight into the HMP, and we can do whatever we want with it, depending on if we're in liver or muscles. In liver, we'll send it out, and muscles will burn it up. So that's the idea here, is that epinephrine or glucagon are going to trigger these depending on the cell type, and we will end up turning on breakdown, turning off synthesis, and working against our counteracting phosphatase. Now, on the other hand, if insulin comes in, insulin undoes everything that, or insulin triggers the activation of protein phosphatase 1. So that undoes everything that PKA used to do. So instead, protein phosphatase 1 is going to uninhibit glycogen synthase. It's going to repress phosphorylase kinase, which means this glycogen phosphorylase is off because you can't turn it on anymore. This is on, and so our net flux under insulin signaling is going to be into glycogen. So that's kind of useful, is that we can switch the behavior just by using either glucagon, which is for low blood sugar, or insulin for high blood sugar. If you have high blood sugar, remember you're filling up your pool fast, and so you need to do something with it. And again, we're going to store it away as glycogen. And insulin can make you do that. Of course, we also talked about how protein phosphatase 1 undoes everything PKA does. It's also going to undo everything it does in glycolysis. And so that means that usually in a muscle, that means that you're going to want to, undo all of those modifications. In liver, it's going to turn on glycolysis. It's going to undo the PKA modifications that turned it off in the first place. So that's kind of the big picture of control of glycogen and glycolysis, hormonal levels, and cell autonomous.